So I'd like to welcome everyone to our 975th monthly meeting. Our guest speaker will be Philippe Reekie of the CFA, and he'll be talking about the potential of alien life existing with an extraterrestrial salt, or at least the signs of a past life. So with that, we'll get off to the agenda, if this would work. There we go. This agenda is pretty different than normal. All the yellow stuff is atypical, so this is our annual meeting, so we will be having the annual reports by the various offices and committees. And the big change here is you'll see we'll have our guest speaker speak first, because this is going to be a pretty long meeting and he's on a tight schedule. So as I mentioned in the beginning, we're going to switch things up a little and have our speaker speak first before the major business section of our meeting. So I'd like to welcome Philippe Reekie from the Center of Astrophysics here. So he's been a communication and outreach specialist from the CFA with a background in planetary science and astrobiology. Philippe's multidisciplinary talk will explore microbial entombment and the potential preservation of extraterrestrial life within salt minerals. So please welcome Philippe.
Right, give me one moment while I connect myself. What's the main cable to connect to the laptop?
So if you just present to Zoom, the video will come through another feed.
Right. So, to share my screen?
Yep. That's okay.
Not too much. This stuff. Hey, thanks for doing that. It's a tremendous amount of work. So,
Does full screen work? Sometimes it flips the wrong program. Looks.
It's fine.
Perfect.
Awesome. Let's get some Zoom bars.
That's easy. I don't have to do anything.
Is this supposed to be?
I'm going to turn down the lights, Sulfate.
Eventually.
Yeah, do you know what that?
I would, or if you're comfortable sharing the whole screen, might get rid of the blue things.
So it's recording. You want that? You want the recording?
I think eventually that top thing should go
That's how we get it
away.
on YouTube.
Just want to make sure.
I don't know how to make that go away.
Well,
I think it'll auto-hide eventually.
They had the highest impact.
okay. So, is that right?
Yeah, sure.
Okay. Is this thing working? Is this working? This is working. Okay. Great. All right. Hi everyone. So my name is Philippe. I work here at the CFA, specifically at the HCO. I'm doing outreach here on behalf of the CFA. So I've been here for 7, maybe 8 months. I'm relatively new. You can probably tell from my accent that I don't come from the US. I come from the UK. And so I'm very, very excited to present my work. But before I present my work, so I'm just going to very, very quickly just introduce myself, who I am, what I'm all about, why am I here at the CFA, and then we're all going to talk about all things alien, specifically my search for extraterrestrials on the surface of Mars. So this one sentence here pretty much summarizes me in a nutshell, from conspiracist to academic to researcher to science communicator. Yes, it is true. When I was a young adult, I was deeply into the conspiracy theory community, surprisingly. I had a very bad, difficult scientific education, and I believed in all kinds of mumbo jumbo, and that is absolutely true. But I always had this like natural curiosity about space science and science in general, and I decided that you know what, I'm going to become a space scientist, and that's exactly what I pursued. As I did a bunch of degrees, and I ended up becoming a researcher of astrobiology. As you can imagine, as I studied these degrees, I completely snapped out of my conspiracy roots. And now I'm a science communicator because I realized that my passion in life isn't research. It's actually my conspiracy roots has driven me to where I am today, because right now, on behalf of the CFA, I actually spend most of my time tackling misinformation and encouraging people. Thank you.
Is your microphone on? Is your microphone?
Yeah, I think it is on. I'm going to put it closer to my face.
Just turn up the volume.
So, is that an option on here?
Pretty good. Advanced audio, maybe.
It's on here.
That should be full power, up to 11.
All right. Come on.
It's kind of dark.
Okay. Great.
There's a hand mic if you prefer.
I think this is okay, right?
Yeah. You can lower it, actually.
Is it too loud now?
That's fine.
Okay. All right. So yes. So now at the CFA, I'm a science communicator, and I do a lot of outreach, and I specifically tackle misinformation, and I know exactly what it's like to be under the claws of misinformation groups, and so that's what I'm doing right now. And I'm very, very excited to see what the future holds for me. And so very, very quickly, what I do here at the CFA, so you know what I'm all about. I've helped resurrect the public observatory nights. So I know a lot of you were heavily involved in this, and I just want to say that I'm very aware of that, and I really hope that we can collaborate like we used to do in the past. So we should totally talk about this, but yes, I do the public observatory nights. At the moment, we've had a bit of a setback with our events team. Some people have suddenly left, and so my plan was to have monthly observatory nights, but at the moment, it's kind of up in the air. So it's looking at like once every other month. And I'm very excited to announce that the next public observatory night, which is at the end of June, is a fully Spanish-speaking public observatory night. So the entire event is going to be hosted in Spanish. We're going to have 2 Spanish guest speakers, and all the astronomers on the roof are all Spanish speakers as well. So that's on June 27th. There are still many tickets remaining. So if any of you understand Spanish, or you know people who are part of the Spanish-speaking community, please get in touch. So that's going to be June 27th. I also do a lot of school visits. I go into schools and I engage with children and tell them why science and space science and astronomy is so cool and so exciting. So I do a lot of that. So, it's supposed to say school visits at the top, I guess.
Can't get that bar to go away.
Yeah, it's a weird bar, but yes, that's there. So if you have any children, you know people who are interested in school visits, then again, let me know. I also do on-site outreach where I host talks here. I do tours around the CFA, the Great Refractor. I work very closely with the museum staff here, the plate staff here, which is just across the corridor. And finally, what happened?
Sorry, I was trying to get that thing to go away. No luck.
And finally, I also do like off-site outreach events. So I engage with the public. Like this is the Great North American Eclipse, and so we had about 4,000 people in the Science Center just outside Harvard, where I was just getting them excited about science. And so that is me in a nutshell. So that's what I'm all about. But now I'm going to talk about the research that I used to work on. So for 6 years, I was working at the University of Edinburgh. And I was working in a laboratory conducting astrobiology research, and it was all about salts. You're going to hear the word salt a lot in this talk. So I was nicknamed the salt guru because all I did for my research was pretty much just crystallizing salts, just like over and over and over again with a bunch of microbes, and I'll explain why. So the story all goes back to here. So this is me as a kid. So I'm actually half Polish. I don't look it and I don't sound it. Very confusing. But yes, I am actually half Polish. I spent a lot of my childhood living in small little towns next to the Baltic Sea. The Baltic Sea, some of you might be aware, is the epicenter for churning up amber. Amber is a remarkable property, and I was fascinated with amber as a kid, and I have a whole massive amber collection back home in the UK. And as you are aware, as many of you I'm sure are aware, that amber has this remarkable property of preserving biology in exquisite detail, far better than how fossils can preserve biology. Because fossilization is a bit of a messy process. You have a lot of heat and pressure, and the whole thing can get smashed up and flattened. But amber, you can get things preserved in 3D, which is amazing. Like this spider right here, this spider is dated it's about 200 million years old, and you can still see the little hairs on its legs, and we can analyze its gut contents to find out its last meal. And this is 200 million years ago, this spider was encased in this amber. And as you know, amber has been the inspiration for a movie franchise, Jurassic Park, the mosquito and bringing back dinosaurs and all of that. So as an astrobiology researcher, I was thinking, is there something like amber that can exist on other planetary surfaces? Because obviously we don't expect to find amber on Mars because we don't have any trees. And so I came across salt. I just realized a lot of my headings are not going to be visible.
Sorry about that.
But it's okay. I can work with it. So salt is an amazing compound. So when I say salt in chemistry, there's a whole family of different minerals. So there's the common salt that we put on our fries, which is called sodium chloride, or we just commonly refer to as table salt. So right here I have sodium chloride, but there are sulfates and perchlorates, all different types of salt species. So salt is an amazing property because when salt crystallizes in an environment, so let's say I get a salty water, I get water and I throw loads of salt in, and I dissolve it in, and I put this salty liquid in a petri dish, and I just let it naturally evaporate. When you let it evaporate, as the crystals grow, it forms these little blobs of trapped fluid inside of the salt. And if there is microbiology present in this environment as these salts are growing, then the microbes can get trapped inside of these fluid inclusions. And when that happens, the microbes can be preserved for millions of years, literally millions of years. This is something that has been documented in the literature countless times. For example, these little microbes right here have been dated to be Permian in age, minimum. Permians, these are at least 250 million years old, these microbes. Question.
Is it still liquid at that point?
Yeah, so these are still blobs of liquid trapped inside of the salt. These are all blobs of liquid, and so these microbes, I should say, so a lot of, oh, you got rid of it? Oh, it's just moved. Oh, it's done now. Hey, there we go. So these microbes, in most cases, they're just remnants of biology. So there's just remnants of some kind of microbe that was sitting there, or some kind of organic matter, like these kind of hairy blobs that are biological in origin, but they're not viable biology anymore. And this is something that's been reported countless times. And it's really interesting because there is one example, which is the most extreme example that I have to share. In the year 2000, there was a paper published where these authors claimed that they got this block of salt that comes from the Alps in Europe. They got this block of salt, and inside they cracked it open and they found this bacillus microbe. And they dated this microbe to 250 million years old, which is not the crazy part. The crazy part is that this was still alive. It was a living microbial colony inside of this salt that was 250 million years old, which is crazy. I should say, highly controversial paper. In the community of astrobiology, a lot of people kind of just dismiss this. They're like, no way, must be contamination and everything. So obviously take it with a pinch of salt. Yes, that is. There's a question. Yes.
Okay. What were the microbes feeding on the salt?
That's a great question, and I should have clarified this. So these guys here, this is a Bacillus subtilis spore. Yeah. These guys, they have this amazing ability to sporulate. So they can expel their juices and they shrivel down into these little seed-like states, and they turn into a spore. And when they are in a spore state, they do not need any nutrients or any source of energy. Absolutely nothing. There's an argument to be made that are they technically living at that point? And so once they're in a spore state, it's essentially a state of hibernation. And they can just sit there, as long as they're protected in some kind of capsule from external factors, they can sit there preserved for millions of years and then be awakened in a laboratory once they sense nutrients or energy in the environment. Yeah.
Do we have to be afraid?
Yeah, do we have to be afraid? I know, I get this a lot. In most cases, no, because when you consume salt yourself, in most cases, any biology in there would be completely harmless and completely dormant. And so your gut is very, very good at just kind of neutralizing anything. And in most cases, if they are in salts, it's kind of trace amounts. So not enough to really infect you or get you sick. But it is wild. It's become very, 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 very wild. So why am I rambling on about salt? It's because the solar system turns out to be a very salty place. Very, very salty place. This is the surface of Mars. This image was taken by NASA's Curiosity rover. And we have these sedimentary bedrocks, and we have these crisscrossing veins. These veins right here, these are salts. And in fact, I have it right here, not the exact one from the surface of Mars, but when we analyze these salts, we think that they are calcium or magnesium sulfates. And this right here is a piece of calcium sulfate taken from the Museum of Science in Boston. And so we've detected this exact salt right here. And in fact, this is also called gypsum. Very, very good at entombing and preserving biology. There's a lot of papers that report microbes found inside gypsum. And so my argument for many, many years is that if we want to send humans, or we want to investigate the surface of Mars and do sample return missions, which we are currently doing, we should really focus on these salts, because if life ever existed on the surface of Mars, maybe this life has found refuge inside of these Martian salts. And that's been my argument for a very, very long time. And in fact, we actually have a bunch of Martian meteorites. We have loads of meteorites that come here. If you're wondering, how do we get rocks from Mars? Big asteroid comes along, smashes into the surface of Mars, creates a huge explosion. Rocks are ejected off the surface, they fly off into space, they fly around space for millions, billions of years until eventually they can land here as a meteorite. And in fact, I have a Martian meteorite right here on loan from the Harvard Geology Museum. This is a teeny tiny little Mars rock. And inside of these Mars rocks, like in this black and white image here, we find extraterrestrial salt. And so this H, this little white blob right there, that's sodium chloride inside of a Martian meteorite. So we know that Mars is very salty, and that's why I think that's very exciting for astrobiologists. When the Curiosity rover drills just beneath the surface, we find more evidence of salt veins. Again, calcium sulfate just growing just beneath the surface. And then the Phoenix lander used its arm to brush away the regolith on the surface, and just underneath, just a few centimeters underneath, they found this slurry of water ice and also various different salt species. But let's put aside solid salts. What about liquid salts? A lot of people always ask me, what happened to Mars's water? Mars is so dry right now. In fact, Mars is far wetter than we think. Mars is actually pretty wet. The problem is that the water on Mars is not very long, it's very short-lived. So any water that happens on the surface, it just kind of doesn't last very long. But water rises to the surface on Mars actually very often. And here the Phoenix lander imaged these little globs of liquid on its arm that lasted several days. And when they analyzed this liquid, it turns out that it's very, very salty. It's a very saturated solution of water. And this is part of the reason why it can survive in the Martian atmosphere, because if you make a water very, very salty, then it changes its properties. It can suddenly withstand the low pressure on Mars, and it can also withstand the freezing from the extreme cold. But eventually it does evaporate and does freeze. But the salts kind of keep it liquid for longer. This amazing photograph right here was taken by the Mars Reconnaissance Orbiter, and it took this image inside of the crater, and these dark streaks right here, this is liquid water flowing down the walls of this crater on Mars. Really, really amazing picture. The Mars Reconnaissance Orbiter then took a spectrum of this water, and it turns out that it is in fact water, and it's also very, very salty. It's a salty, saturated solution that's flowing down these walls. A few years back, the Mars Express took this radar image of the South Pole. It detected this enormous underground liquid lake. This lake underground on the South Pole of Mars. Very, very exciting. Very, very exciting. Could be loads and loads of water underneath the surface. And this is something that I truly love talking about. So Lake Vostok. Has anyone heard of Lake Vostok? Oh, loads of people. Okay, loads of people. Yeah. So Lake Vostok, that's really fascinating. We took this radar image a few decades ago. It's an enormous body of water, and it's living underneath this sheet of ice that's 2.2 miles thick. And a while ago, a group of Russian scientists went above this lake, and they drilled into it. They drilled 2.2 miles, and they got this water, and they sampled it, and they brought it back. to the surface. And we found out, first of all, this lake, we think, has been isolated from the rest of the surface for 20 million years. This body of water has been sitting there in complete darkness and complete isolation for millions of years. But despite that, this body of water is filled with biology. They found all kinds of funky microbes and communities just swimming around in there in complete darkness. And it's really, really fascinating. So like, this is an analog for these underground lakes that could exist on Mars. And also it's a good analog for some of the icy moons in the solar system, like Europa and Enceladus.
So,
Yeah.
how could they exist without either photosynthesis as an energy source or some sort of geothermal?
Yeah. Geothermal energy is a strong contender for how they can support themselves, but also something that is mind-blowing, and I actually found this out not that long ago, that there are some microbes that can photosynthesize in the infrared. Yeah, isn't that super weird? there are some microbes, so these are, what are they called? They're called green sulfur bacteria, and they live around hydrothermal vents at the bottom of our oceans, and they sit there and they can actually photosynthesize in infrared, which is wild. Yeah, question, yeah.
I was going to say, what about gamma rays?
What about camera rays?
Was Lake Vostok salty?
Lake Vostok, yes. It is also a very, very salty environment. And so I should clarify, in case you're wondering, like why? Why is it so salty? The reason for it is because when you have water is a very good solvent, and salt dissolves very readily in water. And so if you have a body of water that's in contact with rocks for millions and millions of years, eventually the components of these rocks, they leach out into the water. And because if there's no hydrological system, like a cycle, to kind of like bring in fresh water and to kind of filter it, then eventually what happens is that this body of water just keeps on increasing its salinity. It just gets saltier and saltier and saltier. And there's no process to remove the salt.
So the assumption is, if this can exist on Earth, it should exist on like Europa and Enceladus and things like that.
Exactly, and in fact this is exactly what we think is happening beneath the surface of Europa. We think that Europa's oceans are very, very salty, and one of the best lines of evidence is that when Europa goes through Jupiter's magnetic fields, it distorts its magnetic fields like quite a lot. And the assumption is that whatever is distorting it is something that is electromagnetically conductive, like a salty body of water. So yeah, really, really cool stuff. So this is one of my favorite images in planetary sciences. This little blob here is Enceladus, one of the moons of Saturn. And Enceladus is filled with volcanoes, but they're cryovolcanoes. These are volcanoes that churn up ice and water, not rocks. And so when Enceladus erupts volcanoes, all this debris and all this stuff, it gets ejected off the surface into space, and it is actually accumulated around Saturn. And this ring right here is called the E ring, like the letter E. Its entire existence is thanks to Enceladus. Enceladus has created this ring of Saturn. Now, the Cassini spacecraft, just before the end of its mission, did some like crazy maneuvers, because the researchers were like, okay, it's achieved most of the things that it's achieved. Let's now kind of like do some wild experiments. And so the Cassini spacecraft actually flew through this ring and opened up its belly and absorbed the stuff into its guts, and it analyzed it using mass spectroscopy. And it turns out that this ring is mostly made up of water ice and various different salt species. So we actually have sodium chloride and sulfates in orbit around Saturn and make up this ring. So the reason why I'm talking about this is because I want to kind of demonstrate just what a salty solar system we live in. It's very, very exciting. So I'm going to very quickly just briefly pitch what exactly was I doing in this lab for many, many years. So I was creating different salty solutions. I was using like chlorides and sulfates and perchlorates. You just add these compounds into water and make saturated solutions. I was cultivating microorganisms. I was using these three guys. Halobacterium, which is not actually a bacteria, it's an archaea. So it's just a weird name, but yeah, Halobacterium is something that is it's an extremophile that can survive a very high concentration of salt. Marinococcus is a type of organism that can survive very high sulfur concentrations, and Crucochidiopsis, my favorite, is an organism that we have evidence that this organism has existed on Earth for at least 2 billion years.
Wow.
So it is a candidate for like early life on the surface of Earth. So I was cultivating them, and I was mixing the microorganisms with the salty solutions, putting them into a petri dish and just letting them naturally evaporate over time. And as they evaporated, they crystallized. Salts are also referred to as evaporites. So it's evaporites because they kind of crystallize from evaporation. And so as this was happening, I was trying to get my microbes like entrapped and entombed inside of these salts, like inside of the fluid inclusions. And then I would observe them, and then my main kind of aim for my research was what salts should be prioritized for astrobiological investigation, because like I've said, that salts is a whole family of groups, many different species on the surface of Mars. And so which one should we prioritize for our sample return mission? And here we go. I'm just going to quickly show you some fascinating pictures. So this is a tiny piece of sodium chloride in my lab, and inside you can see these little rectangles. These are all the fluid inclusions. I had a very fun Friday evening counting all of them, and I got to about 5,000 and then gave up. There's a lot of fluid inclusions in here. And so every single one of these fluid inclusions is filled with biology. You can't see it because it's very hard to photograph microbes in plain polarized lights. So what I did is I stained the microbes with a special dye that made them glow green under UV light. So if we switch on the UV lights, looks like something from the Matrix, but it is actually the same piece of salt. And so here we have the fluid inclusions, and now you can see the microbes that are fluorescing inside of them. There wasn't a single fluid inclusion inside of this tiny piece of salt that didn't have evidence of biology.
How did you get the dye into the salt?
So the cells were dyed first individually,
Oh, you did this before. You did this
Yeah.
before, and you dyed. Which fluorescent dye did you use?
So this was, let's see if I can remember it. It was called, oh my God, what was it called? It was gold something.
Okay.
It's called gold something. I can't remember what it's called, but yeah, has the word gold in it. Yeah. So
So these were basically dead microbes at this point because you dyed them.
These are alive.
They were alive even after they've been.
These are alive. So this is a DNA binding dye, and in order for them to fluoresce, they have to be living.
Okay.
Because as soon as the membrane kind of breaks down, then the dye stops working.
Very good. Okay.
Yeah.
So this is a non-destructive dye.
Yeah, non-destructive dye, and so they were glowing, as you can see them. And then actually I have a little video so you can see what I was looking at. You can see them just kind of wiggling and jiggling inside of this fluid inclusion, and they're actually wiggling not because of they can't move. They're not motile. They just kind of sit there, and they don't have the ability to move. But they're moving because of something called Brownian motion. Yeah.
Einstein 1905.
Yeah. So. Yeah, and this exact sample is still in my lab at the University of Edinburgh, and it will sit there for a very long time. And I'll get back to that in a moment. You'll see what I mean. But yeah, and then if we compare them to like other salt species, so sodium chloride was a clear winner. It has this amazing ability of entombing microbes and preserving them very, very well, unlike other things like sulfates. And the reason for that has to do with some really complicated mineral physics. It's because the way sodium chloride crystallizes, it kind of crystallizes from the outward going inward. And so it can like scoop up the biology and put it inside of it. And the biology just sits inside of it, and it can just sit there for a very, very long time, potentially millions of years, preserved. Unlike in sulfates, that didn't really work out. But anyone who knows the chemistry of what's going on Mars well will know that there is one salt species that is the most abundant, and it is perchlorates. Perchlorate is a type of salt that is by far the most common salt species on the surface of Mars. It is so common, in fact, that so here on Earth we have perchlorates. They exist naturally in our soils, and on Mars the concentration of perchlorates is 10,000 times greater. The highest concentration of perchlorates here on Earth is in the Atacama Desert, which isn't a weird coincidence. The Atacama being somewhat similar to Mars, like seen as like a good Mars analog. Perchlorates synthesize themselves in an environment that's very arid, very dry, arid environments that has UV radiation. And so what's happening here is that the UV radiation is smashing the rocks and the soils here, and is synthesizing these perchlorates in a very, very high concentration. And so if anyone's going to study biology in this, potential biology in this environment, you really have to study perchlorates. And so for about 2 years, I was looking at sodium chloride. But then after that, for the next 4 years of my research, I got a new funding grant to specifically study and investigate perchlorates. That's where I'm now going to talk about perchlorates for the rest of this talk. So the problem with perchlorates is that they are really, really nasty for biology. They're not only nasty for biology, they're nasty for our potential future astronauts that can go to Mars. This is a huge problem that we have to navigate through. So perchlorates, we use them for rocket fuel. They're extremely explosive. They're powerful oxidizing agents. They are weirdly soluble in water. And I say weirdly because, you know, remember when you're in science class and you're adding sodium, like table salt to water, and you keep on adding it, mixing it in, you dissolve it until eventually it stops dissolving. The salt just doesn't dissolve anymore. It's because you've reached a saturation point in that water. But if you keep adding perchlorates into water, it just keeps on dissolving. It just keeps on going. It keeps on dissolving. You keep thinking that, like, no, it must be saturated. But no, it just keeps on going, keeps on dissolving. And the reason why this is dangerous is because you can make some truly toxic, concentrated solutions of perchlorate that can literally corrode human flesh. Really, really bad. But also, perchlorates are very weird because they kind of, like the molecules, they mess around with water. They make water pretend as if it's like under 2 gigapascals of pressure, which is also like a really strange property. But basically the summary is that perchlorates are really bad for biology. So this is a huge problem. And this image right here just demonstrates how nasty they are to biology. These are Bacillus subtilis cells, like the Bacillus before, and I just dropped them in some perchlorate solution. And within seconds, the cell walls started effervescing, and they got all frothy and gross, and within minutes, their guts just kind of exploded all over the place, and they all died. So cells were very, very unhappy in perchlorates, and so the challenge that I had is, like, how on earth am I going to try and, like, entomb microbes inside perchlorates when perchlorates are clearly really horrible? So I was thinking a lot. And as any good researcher thinks, the best place to think is down in your local pub with your friends over several pints of beer. And so I was at a local pub with my friend, he's a cardiologist. He works in medical sciences, and he mentioned glass to me. He was like, "Have you considered glass?" And I was like, "You probably had too many beers at this point. I don't know what you're talking about." And he said, "Well," he said, "No, because he works in cancer research." And he said that in his lab, what they do is they take cells and they preserve them in extremely cold glasses, and they can be preserved for decades, even hundreds of years, these cancer cells. And so it got me thinking, I was like, what if I can make salt glasses instead of salt crystals? And that was the inspiration of the next 4 years of my work. And so I just completely dropped all these minerals like this. This is obviously a salt mineral, so it's a crystallized form of salt. And I was like, why don't we start looking at salt glasses? Because this kind of makes sense. This kind of makes sense. Because the process of crystallization can be actually quite damaging to biology. Because when crystals form, the whole process kind of makes atoms rearrange themselves in a particular configuration. And this configuration, like if you have ice crystals, it can often like puncture and rupture your cells. And the easiest way to demonstrate this is this highly sophisticated experiment I did in my home, where I took some frozen berries out of the freezer and just let them defrost naturally. And all of you will know that when you let something like berries defrost, then a pool of juices just come out. And the reason why this happens is because all of the cells inside of this berry, they were frozen in the freezer, and so ice crystals formed inside of these berries, and it's kind of ruptured and split the biology. And so when you thaw it, its juices just kind of pulled out. And this is the problem with crystallization. But in glass is essentially like a liquid that's frozen, like on an atomic structure. So like a glass and a liquid on an atomic scale, very, very similar. It's just one is a liquid, but one is a solid. Because you have this like disorganization of atoms, but it means that when you have glass formation occur, you can actually encase and entomb cells in perfect exquisite detail like you do in a block of amber. Right here we have, this is a human liver. Human liver, on the left side, it was thrown into a freezer at minus 80 degrees Celsius. And ice crystals formed, completely rupturing and destroying the soft tissue of the liver. And on the right-hand side, we have the same sample, liver tissue again, but it's been vitrified. So vitrified is just, if you haven't heard of it, the word is a fancy word for glass formation. So if you want to turn something into a glass, you vitrify it. So this was a vitrified piece of liver, and it stayed in perfect condition.
How do you do that?
Yeah.
How do I do what?
How do you vitrify something like that?
So something like this. So in biomedicine they use special alcohols, which they saturate in a special alcohols. There's one of them called, I think it's called glycol. And they saturate in glycol and they plunge it into a very cold bath to immediately vitrify it.
Okay, so the glycol prevents the growth of crystals.
Yes.
Okay.
Yeah, so it's done under like very controlled environments that can stop the process of crystallization occurring and just vitrify them. Yeah.
You say glass, and I think a big oven and melting window glass and stuff like that. Are we talking the same kind of glass?
Yeah. So a glass in chemistry is essentially just imagine the best example is to imagine it like a liquid, but it's solid. So imagine like a glass of water, but instead of like it swishing around, it's completely solid.
Right.
And so glass is kind of like that on an atomic scale.
So how do you embed this in the glass with those temperatures that are involved to get the glass?
You can
Do you liquefy the glass? You can't liquefy the glass to put it in there because
rate of
you burn up your.
crystallization.
Yeah, it's
Go
so
faster than it crystallizes it.
in biomedical sciences, I'm not entirely sure how this process is done. I will touch on what I did for forming the glasses, which might help answer that question. But in terms of like this for the liver, I'm not entirely sure. This was done by biomedical scientists, but yeah, I'll get back to that in a couple of slides. Yeah, I know. Didn't expect that, did you? There we go. The good old tardigrade. Does anyone not know what this is? Does anyone over there maybe? So quickly just summarize the tardigrade if anyone doesn't know. Also nicknamed the water bears. Tardigrades are very famous in science because we've realized that these tiny little microscopic animals can survive all kinds of extremes. We can expose them to enormous amounts of pressure, high pressure, low pressure, high temperature, cold temperature, acidic environments, alkaline environments, and they seem to be just fine. NASA put them in the space environment and they were just like, yeah, we got this, we got this. So tardigrades are fascinating, and the reason why they're here is because I found out something so cool about tardigrades. So when tardigrades sense some kind of threat and they feel unhappy, the reason why they can survive all kinds of extremes is because as soon as they sense a threat, they eject all of their juices from their body and they shrivel down into this tiny little seed state, and it's called a ton state or a cryptobiotic state. And when this happens, they then vitrify all of their organs. Their organs turn into glass. And so that was like, whoa, there we go, that just proves my point about how glass can be a very effective way of preserving biology. And the tardigrades have managed to crack this just through evolution. They can actually vitrify their organs, which is a very, very rare thing in the animal world. So going back to perchlorates, I was like, okay, well, what if I can make a perchlorate glass? Let's consider that. At the time of me doing this research, there's one paper in the entire world that has turned perchlorates into a glass. Was there a question?
No, I was
okay. And it's this paper here that was published in 2014 where this guy was like looking at perchlorates and was looking at like, well, can we vitrify them? And the answer is yes. You can vitrify perchlorates, and it does it in the most weirdest way possible. Because generally speaking in nature, the way how glasses are formed, a classic way of how they're formed is, let's say you get like a lava and you throw that lava into really cold water. Then the lava can immediately turn into a glass, like a volcanic glass. That's how volcanic glass comes about, because you get a lava that's immediately quenched with extremely cold temperatures. So that's typically how you form glasses. But with perchlorates, it's very, very different. You've got temperature and time on the other side. With perchlorates, you simply just cool them down. So I have like a saturated solution of perchlorates, and I'm cooling it down, and around minus 120 degrees Celsius, they just vitrify. They just vitrify slowly. And it's really, really peculiar, very, very peculiar. So what happens is this liquid, as I'm cooling it down, its viscosity is increasing. It's getting thicker and thicker and thicker, until eventually at minus 120, it's so thick that you can no longer consider it to be a liquid, and it's now a solid. And at that point, you have vitrified your perchlorates. And this is very promising for the surface of Mars and other places in the solar system, because the chances of a perchlorate just suddenly vitrifying from immediately being quenched is quite rare. But in this case, you can have actually demonstrated that if you have perchlorates existing on the surface of Mars, if they can just cool down near a minus 120 degrees Celsius, which is a common temperature around the poles, then you can just have natural glass formations of perchlorates on Mars, which is wild.
But don't they melt?
stay vitrified if it warms up again?
If you warm it up, then the viscosity decreases, and then eventually it goes back into like a thick liquid. You keep warming it up until it gets more of a runny liquid. And so it's this weird transition of like liquid to thicker liquid until eventually it's so thick it's no longer a liquid, it's now a solid. At that point it's a glass, and then you can undo that.
One of the things you pointed out earlier, though, is that perchlorate's extremely poisonous to most things. So when it's a liquid, it would kill anything that was in it, and therefore there would nothing be around to vitrify.
Exactly. So this was part of the challenge that I was exploring.
Do the perchlorates lose any of their chemical constitution as they get cooler and then warm up?
What exactly do you mean by constitution?
What can I say?
Kind of like an acid etches more at a higher temperature. Do they become less nasty at colder temperatures, or the rate of being nasty?
That is a really, really good question, and I don't know. I actually don't know if it does, but perhaps some of the results that I will briefly show you, they might indicate something that might indicate something for that question. Yeah.
You know, it's a really interesting idea, because as the liquid becomes thicker, it might have less of an effect on the cell walls.
Yeah, exactly.
And that's how it could potentially capture the biology.
Yeah. So this next slide.
Critical point.
Yeah, this next slide is kind of related to what you're saying. So there's a bunch of papers that are tested the toxicity of perchlorates with various different microbes. And generally speaking, there are a lot of papers that suggest that like if your perchlorates are colder, then the survival rates of your microbes is increased. And so in this case, everything here died, all the microbes died. But there are cases like this, the blue line at the top, which is at negative 30 degrees Celsius. The survival rate actually lasted several hours. So the microbes were able to kind of hang on for a few hours before they eventually all perished. And so this is also just kind of basic chemistry in the sense that if you have something colder, it can kind of like reduce the chemical reaction effects of your materials. And so what I was doing for 4 years is that I was taking this concept of perchlorates, knowing that they're highly toxic, realizing that there's a potential to turn them into a glass, and I was like then throwing in a bunch of microbes to see if I can entomb them inside of a block of perchlorate glass. And the main reason, the main research question I had was, is this process lethal to my microbes? Could microbiology potentially survive this process of being vitrified in a toxic compound like a perchlorate? And the way I was doing it is I was using this guy. This was specifically designed for me. It's a custom built, I just nicknamed it the cryo stage. And it is something that hooks up to liquid nitrogen tanks, and it hooks up to a microscope. So you can put a sample in it, you can chill it down to minus 150 degrees Celsius, and then you can observe it under a microscope. So I was using that, and I was also using this guy. This costs about $100 million. It is a cryofib SEM, a state-of-the-art one, which stands for Cryogenic Focused Ion Beam Scanning Electron Microscope. Quite a mouthful. Now this guy right here, a very, very complex, sophisticated piece of machine, they were charging me, or my grant money, what was it? Something like $300 per hour. And it took 3 hours to turn the thing on. Yeah. So, absolute scam. But yeah, so this cost me a huge amount of money, and so this is the tiny little stub that I would fill with my perchlorate, saturated liquid, with the microbes all inside of here. And then they'll get plunged inside of this chamber where they would get vitrified. And now, very, very quickly before I show you some of my perchlorate glasses, what microbes did I study? Well, I already told you that I mentioned Bacillus subtilis. So Bacillus subtilis, this is a photograph that I took myself in the lab. Bacillus subtilis is a very interesting microbe. There's a reason why astrobiologists are obsessed with this microbe, because it is a very good model organism for astrobiology. It's a very good model organism for abiogenesis, for early life on Earth, and it's also considered a good candidate for panspermia, which is this wild hypothesis that life could potentially be seeded from planet to planet, like in this artist depiction right here. Oh, what's that?
We're being invaded.
Being invaded by the microbes.
By telephones.
Is this so from Archaea up to your Bacillus that you're using, are we still expecting, whether it's photosynthesis or chemosynthesis or whatever, are we still expecting, though, the carbon, hydrogen, oxygen for, at least the glucose input for cellular respiration, or?
For these microbes, these microbes specifically?
So for energy production, right?
Yeah. So,
I mean, are we still expecting that carbon, hydrogen, oxygen chemistry?
yes. My research was, yes. There are some people that are exploring alternatives, which is a really interesting field, but not me. I was just using the kind of the generic understanding of what we consider to be necessary for life. But fun fact about panspermia, there's a university in the UK called the University of Kent. The University of Kent has this massive piston gun, and they published a paper on how feasible is this whole concept of panspermia, but they're specifically looking at fossils. They're looking at microscopic fossils in rocks, and they're shooting them with this big gun to kind of recreate an asteroid impact, and then they were bombarding the samples with radiation to recreate the space environment, and then they took that sample and they loaded it into the piston gun, and then they shot it into a surface to recreate a planetary re-entry onto a surface. They basically try to recreate the whole process of panspermia with microfossils, and it turns out that there are certain fossils that can survive this concept. They can survive planetary ejection, the space environment, and planetary re-entry. And so one thing I always find really fun is that one day, this is a complete side note, one day we can actually find fossils on the surface of the moon. Because with the fact that we have loads of rocks from the moon through planetary ejection, it means that the moon very likely has rocks from Earth. And so one day we can pick up a rock on the moon and be like, this looks a bit weird, and we can crack it open, we can find evidence of biology inside of this moon rock, and we realize that it doesn't come from the moon. This is biology that comes from the Earth, which is kind of fun. But yeah, so I was looking at Bacillus subtilis for all of these reasons. So it's a really good model organism in microbiology. This is a little sample of my perchlorate glass. It's a tiny little sample, and then here what you're seeing, these are the Bacillus microbes inside of this sample. So these microbes are just stuck in this block of glass, completely motionless, and they can't do anything about it. And they're probably very unhappy, but there you go, they are there. So this is, I was looking specifically at the magnesium version of perchlorate. So perchlorates I didn't mention this earlier, but you can find like magnesium perchlorates, there are calcium perchlorates, sodium perchlorates, and all three of these species have been detected on the surface of Mars. This is a fun one. So this is a sample of magnesium perchlorate, and you can see these crystals, because this is a crystallized version. And on the right-hand side, which I'm about to show you, the exact same sample, both of them are filled with biology. Exact same sample. And that is my perchlorate glass. The difference is that this has been crystallized and this has been vitrified. And so you can immediately see how this seems to be a far more preferable medium for encasing biology than something like that, where you have these sharp, jagged spines that can penetrate and rupture your fragile biology. And so this was filled biology, so that my next objective was to try to crack this open and try to find this biology, which was really hard. It took me many, many hours and cost me thousands and thousands of dollars. This is a similar image like the one before. This is a little wedge. So this is the surface where you have all this condensation, and then I use a laser to kind of penetrate it and look at the internal structure, and you can see the very smooth surface over here. But still no evidence of biology. But here they are. I finally found them after many hours of digging. And you can see right here, this is very interesting because my cells are rod-shaped. Bacillus subtilis are rod-shaped, and everything here, nothing is rod-shaped. These are all spherical. And the reason why they're spherical is because they turned into spores. So earlier I mentioned how this is how they survive these extreme environments, is that they sporulate. They turn into spores, and then they can just sit there for potentially millions of years and be preserved. Over here, this is one of the spores that looks like it's been ruptured. This is a process that we call cell lysis. It's when the cell wall just kind of breaks down and its guts kind of leak out. Yes, question?
So you proved that the perchlorates are not damaging the spores or the bacteria?
Yeah, so in this case, the spores were all intact, or mostly intact, but the evidence of these spores was, looking okay, and part of the reason for that was probably because this is under an extremely cold temperature. So this temperature right here was around minus 150 degrees Celsius. And so at this extreme level of coldness, then that's probably how they can survive, because the chemical reactions that could rupture these cells is somewhat reduced and negated, but not entirely so. As you can see right here, it's actually ruptured right here. But I finally found my cells, and there were millions of them inside of it. So it was challenging to find them. But I finally found these spores, and if you take a closer look, you can see them cemented in this kind of waxy substance. And this is
why were the rods turned into spores?
So the rods, they start off as rods when they're metabolizing. So that's when they're kind of happy and doing their thing. And when they feel like a scent, like some kind of danger or like a lack of nutrients, that's when they turn into a spore. Because when they're in a spore state, they are highly resilient and they can survive a lot of extremes. And so you have this kind of like cemented image right here, which is really cool. I then took them out and I put them back into water. I kind of washed them, I put them back into water, and these spores then came out of their spore state and they became rod-shaped again. And this image right here is using something called TEM, which stands for Transmission Electron Microscopy. And I took these images. Now this is really, really weird, because you see right here, you have this cell and you've got this kind of like this indent right here. This is called a mid-cell division. And generally speaking, when you see that in a cell, it means that they're happy, because this is them reproducing. So the cells, they split in half, and then there's two versions of them, and that's how they reproduce. But its buddy has just exploded next to this guy. This guy, I actually saw this happen in real time, and I was really annoyed that I didn't have my video footage filming. So I was looking at this cell, and then suddenly it just popped. And all this is kind of like this liquid just spewed out of its cell. But it's really weird because, again, this cell has this mid-cell division, implying that it's kind of a happy bunny, but it's not. It's trying to reproduce while simultaneously exploding. And I think this is a premise for someone's PhD, just this one image, because I don't fully understand what's going on here, and neither did my supervisor. But yeah, here they are. And then what I did was I then plated all of my cells, and because I wanted to see if could they grow on an agar plate, and the short answer is yes. This process of taking cells, putting them into extreme cold, and vitrifying them into a block of perchlorate, I demonstrated that it is not lethal to them. This process is not lethal, at least not for Bacillus subtilis. And earlier I did very briefly touch on where can we find perchlorate loss? To date, we've never ever.
The thing with an exploded cell, is it possible that that slide still exists? Okay, it's possible perhaps to see if you can see the circular chromosome and see if it's still intact.
Yeah.
Because bacteria have circular chromosomes, and of course, if it was dividing, you would have them coupled, so you'd have actually be able to see maybe the topoisomerase separating the the chromosomes.
No, so it's a very interesting point, and I'm no longer working in this, but we do have the people who are
But
currently.
that one is still there.
Yeah. So all my samples are still in my lab, and there's a new team of researchers who are continuing this line of work,
Okay.
which is very, very exciting. And so we've never detected perchlorate glass on Mars, but we know that perchlorates are extremely common. They're all over. Yes, question?
Yeah, I would have thought that if you put a microbe in an inclusion where it's surrounded by salt, the salt would suck all the liquid out of the microbe and kill it and probably destroy most of the interesting structure too, but apparently that doesn't happen.
That's it. So that does happen. That's called osmosis. It's when that's how a lot of these cells die. And so remember that picture where I showed the microbes that were effervescing on the sides. Their cell walls were just breaking down. That was the early stage of them just about to die. And so that process does happen with perchlorates. But the difference is that my microbes were only in perchlorates under really cold temperature. And so that cold temperature, my hypothesis is that cold temperature was slowing down that process of osmosis to keep them alive for longer.
Sure.
Yeah. So we've never detected perchlorate glasses on the surface of Mars, but there's no reason why they can't exist. And so, especially near the poles, I have to apologize. Because I'm British, I'm using still Celsius. I'm still getting accustomed to Fahrenheit. But yeah, near the poles, there's no reason why they can't exist. So I should clarify. Do I think that the first detection of alien life is going to happen in perchlorate glass? No. I do not. I think there's far more better mediums to be investigating. This was just kind of like a weird, wacky project that I was throwing a bunch of money to kind of investigate to see if it's like feasible. And so I guess like my research did demonstrate that it is feasible. Do I think it's like a good kind of indication to how we can find alien life? Probably not, but I think it's still very interesting. Something to consider.
Send a probe to the ice cap.
I know. I really wish we send more probes to the poles, because I think we'll find some really spicy stuff happening at the Martian poles. This is my last slide. Wow, it just flew by. But my last slide is this. So this is this is a fun one. I published this paper along with my colleagues in my old lab, because there's me with I don't know what I was wearing on my feet. That was my fashion style back in the day. And we published this paper, and in this paper, I proposed the world's first 1 million year long experiment. And this is 100% real. And in this, you'll find my proposal for how I'm going to get my Petri dishes with these salts, all of them filled with microbes. And what I want to do is I want them to be monitored and analyzed at certain increments for the next 1 million years by our future science ancestors. And the funny thing is that the Museum of Scotland, the National Museum of Scotland in Edinburgh, they called my research lab up and we came across this paper. You know what? We want to actually have an exhibition of your work in this museum, which is going to be there. It's going to double as an exhibition, but also a research lab, and it's going to be there for the next 1 million years. And yeah, so that's very, very exciting. And so, yeah, very, very exciting. And so, yeah, thank you all for listening. So, I can take some questions. Yeah.
Early on in your talk, you showed a bacterium 3 nanometers long inside a salt crystal that I believe you said it was 1 or 2 billion years old.
Oh yeah, the controversial paper
You
one.
said it wasn't dead. Has that been extracted and did it come back to life with water?
Yes, so those microbes were extracted from that alpine salt, and they were in a spore state, and then they suddenly came out of their spore state and became viable rod-shaped cells. Yeah.
So you actually found, so 2 billion year bacterium came back.
Quarter of a billion years old.
That's very impressive.
Yeah. But I also want to just remind that it is very controversial, this paper.
Follow-up question. a few years ago, there was some Martian meteorites found in Antarctica that had bacterial-like inclusions.
Yeah.
Does anything more come of that?
Yeah, that's a great question. So there was a meteorite that was found in Antarctica. I weirdly remember its name. It is called ALH 84001. Don't know how I remember that, but it's a very famous meteorite because inside of it, they cracked it open and we realized it comes from Mars. And if you're wondering, how do we know if it comes from Mars? Because Martian meteorites have little trapped pockets of gas inside of them. When we analyze the gas, it's chemically identical to the Martian atmosphere, which is strong evidence. And so inside of this meteorite that you're referring to, we found these little structures that look like little worms, so look like little fossilized worms, and everyone was very excited about this, and it made front page news that we found alien life on Mars. In terms of like anything new that's come about, I haven't heard anything come about. I know that for the most part it's been mostly dismissed as being biological, and the main argument for that is because these little structures are so tiny. They're so tiny to the point where people were making an argument saying that this structure is smaller than the bare minimum in order to have a biological thing. So because the structures were just like a few molecules large, but in order to have an actual biological thing, you need to have quite a lot of chemistry, quite a lot of chemicals and molecules that all come together. And so that was one of the reasons. And then we also realized that similar structures are formed in nature, and we think that they are carbonates. We think that they are carbonate precipitates that happen in the rock.
In the grand scheme of astrobiology, what sense of importance does the discovery of these microbes on Europa, Enceladus, or any other planet out in the universe have on society? Does that mean that if these are discovered on Europa and Enceladus, could this be a precursor to the formation or evolution of life on other planets?
I believe so. I mean, it's very. I mean, it would mean a lot for us as a species. In fact, in my old lab, we had a similar group like this that would come together once a month, and we would host guest speakers. And we once had this astrobiologist come in, who was he was an astrobiologist who was a philosopher, and he gave a talk about like what it means, like philosophically, to us as a species if we were to find these types of microbes on Enceladus or Europa and big questions like that. But yeah, I think it's a very, very fascinating time to be an astrobiologist, and I truly would think that, well, at least I hope that in my lifetime we will find a strong detection of alien life. Really hope so.
But we won't get invaded by these microbes.
I don't think so. I think we're all right. We'll be all right. Yeah.
Sorry, a side note, sorry, this is not astrobiology, but vitrification, do you think that's actually how they try to freeze human organs in the end?
Yeah.
And they're talking about like to have people
Brought back to life.
brought back to life after a century or something.
That is exactly how they freeze human organs. So human organs, it's really important that when you preserve a human organ, you need to make sure you don't form crystals.
Oh.
If you form crystals, the human tissue gets ruptured and destroyed, and so they're normally controlled in a very special environment, and they often use glasses to kind of embed it and entomb it into a block of solid material that is then preserved in 3D. Yeah.
Okay, so this means you have to have a glass, like a silicon oxide materials in that.
Yeah.
Okay.
But you can't crystallize a human body in a chlorate salt.
Definitely not recommended. How am I doing for time?
Should probably wrap up soon.
Probably wrap up. Okay. Should I take one more question or just
One more time.
one more question? Oh, over there.
Okay, so Perseverance is on Mars now gathering samples in Jezero crater.
Yeah.
And it's pretty well known what is in each tube.
Mm-hmm.
is there any chance that some of those samples we already have gathered or about to gather might contain salt,
Yeah.
high salt environments that could
So-
serve like?
Last time I checked, so Perseverance is creating these cores. They're just drilling up and creating these cores and then depositing them on the surface. And they're just kind of sitting there waiting for a future mission to go along, collect them, and return them to Earth. And so they have, to my knowledge, they have not extracted like one of these veins, you know, those veins of calcium sulfate. They have not extracted like a block of that, but they have extracted some sediments that do contain a high concentration of salt inside of them. Yeah, so it's very, very exciting. So as soon as those samples come back, it's going to be a very exciting time for planetary geology and astrobiology. So I'll wrap up there. I want to quickly add that I have to sadly run because I'm on a bit of a time constraint. But I have a bunch of business cards right here, which I'm going to just leave here. So if you have any questions about anything, any of my research, or anything to do with public observatory nights, or anything in general, just drop me an email. Drop me an email, and we can chat. Okay.
Thank you so much.
This
Minor samples too.
White sands.
Okay, yeah, good. All right.
Thank you so much, and I
Yeah.
hope we do collaborate on the observing
Absolutely, I
nights.
hope so.
All right. Maybe 1-2 minute, stand up, get a drink, and then we'll get down to business.
Well, Glenn came and went because he's not feeling well. And when he knew that our little part of the show would be later, he felt that it was in his best interest to get home. So I'm going to bore you with just me. So we think this is from Sal.
Oh, come on. Oh, wait a minute, I wish I thought of that.
And Glenn wanted to keep with the Aurora theme since we were all treated to quite a display just recently. So you're going to see a lot of Aurora stuff tonight. But if you could do the next slide, so this was taken from Facebook by Carrie Lynn and sent to Glenn. And the next slide also shows that if you did go out, it's best not to put your eclipse glasses on. And so the source of you will never forget the night of May 10th and 11th.
The 12th.
And even the 12th.
It's the 9th, the 9th was the meeting.
Yeah, the 9th is the meeting.
Those explosions happened before the meeting because.
That's right, it takes a while for the.
Well, no, I pulled them off the Solar Dynamic Observatory before I came here. They're still on there.
Well, it takes a while for the particles that are expelled from the sun to reach the Earth, but this was the source, which is actually wheeled around the backside of the sun and is back in the front. Not in its original glory, but you can still see this sunspot, 3664, I think.
That was, they said that this was bigger than the sunspot during the Carrington event.
Yeah, or at least certainly comparable to it. It was, there was some post that had the two sort of superimposed. Big active sunspot region, and what we benefited from was a wonderful display of the Northern Lights. And so what?
Can I just comment?
Sure.
On that picture, because I took it. And just go back. That's a white light picture. It's taken through a neutral density beta solar film. But I'm personally amazed at this detail. And people ask me, how do I get that detail? I say, I take a lot of videos and I hope one or two of them come out well. So this was.
Sure.
This is one of them, but
Well,
that's the type of resolution.
Daytime observing, typical daytime observing, the scene is terrible.
Right,
Seems
normally
awful.
it's awful, but this was one of those dumb luck things, the morning after our meeting, and then it was that night that everything happened here.
Yep.
What telescope did you use?
Huh?
What telescope?
That's a Celestron Nexstar 6-inch SE. So it's a 6-inch and the filter is the full aperture filter that's put on,
Was it an ND5 or an ND3A?
it's an ND5.
Okay.
So this was taken on May 10th?
This was taken the morning of May 10th around 11 o'clock.
Oh,
All right?
And so what Glenn did was he asked the membership if they were,
Yeah, we did.
if they had a picture they really enjoyed, an image that folks took that they wanted to share. And he put them in sort of a chronological time order, just to see what people were actually seeing. And so we won't spend a lot of time on these because the meeting is gonna run really long anyway. But Bruce took this one from York, Maine at 9:27 in the evening. That's about the time I went out when I first decided to go out and see what was going on in the sky. There seemed to be two nice bursts of auroral activity here in New England. The early one was from about 9:30 till about 11:30. And then later on, in the wee hours of the 11th, there was another burst. So we can click through these slides fairly quickly. Bernie Volz took this from Center Harbor, New Hampshire, a little bit later, 10:06, a nice corona. That's very nice. Sort of looking right at the magnetic field lines. Jonathan Morris from Chelmsford, Mass. at 10:15. James Clem from Belmont, Mass. at 10:15. Next slide is Alan Slisky from Lincoln, Mass. at about 10:15. I think this is the slide that, I'm not sure, Alan, you actually put the time on there, but what Glenn did was he took out his old-fashioned planisphere and he set it, so and there's the Big Dipper, right? And he set the planisphere, so the Big Dipper sort of mimicked the position. And he was like, oh, it's about 10:15. So that's how Glenn got the timestamp for this one here. So next slide is a little bit later. Nazareth took this from East Arlington at 10:41. There was a little bit of cloudiness in the skies there. Next slide was Bert Halstead from Belmont, about 10:15. Some really nice aurora display there, and on we go. Doug Paul from Bedford, Mass. at about the same time. This was from Jeffrey Alexander from Arlington, Mass. at 10:45. John Bishop at the Clubhouse, about the same time. Mark Helen from Ipswich at 10:46. Now Mark had a nice clear view of the horizon here. And when you look at aurora displays like this, the green is actually taking place lower in the atmosphere than the reds. Sure. And so you could almost calculate how far away this aurora was from Ipswich, because you know the altitudes of those things. And so you could do a little SOHCAHTOA, like the stuff you never thought you'd ever use again from high school math, right? And you could actually calculate how far away that aurora display was. They're hundreds of miles away. Pretty cool.
See the reflection in the water.
Yeah, I like the reflection in the
I have a shot
water.
from that from 1981, at almost the same place, reflection of aurora in the
Nice,
water. Pretty
very
cool.
nice. Bill Dwayne from Westford, a little after 11 o'clock. Mike, I'm not even gonna try and pronounce your last name, Mike, but from Lancaster, Mass, 11:36. So a little bit of cloudiness moving in there. And for those of you that stayed up late, like me, actually took a nap. I watched the early show, and then I went in and I put the TV on. For when it died down, I put the TV on, knowing full well that I would fall asleep, but also knowing that nature would wake me up a few hours later. And so I got up, and went out to the kitchen window, I looked out and I went, oh God, it's on again, I've got to go back up.
So at our age, doesn't nature usually wake us up every night?
A few hours, gives you a few hours of sleep, and then you wake up, and then a few more hours of sleep if you're lucky. So I took this from my backyard in Framingham. This was taken with an iPhone 14. Until this night, I didn't realize the iPhone would do this. And I just figured I'd hold it up and see what it did. And it's actually got like a night mode that it just kind of clicked into. And these are 3 seconds, handheld 3 seconds with an iPhone. Pretty colors, really delicate, beautiful colors. And then one more, since nobody else had sent Glenn anything from after midnight. This was a nice shot of the corona. Now you can see Vega is the bright star up here, and there's Lyra. And this is Alpha Ophiuchi, that's Alpha Hercules. So this corona was about 25 degrees south of the zenith. And it was really beautiful, really pretty. So I've got about 250 images that I just snapped. I know we've been talking about me getting into nightscape photography, but there was no time for that.
No.
I was just shooting with the iPhone, and they came out great.
I think we need some kind of alert system for these once-in-a-lifetime events in the future.
You know, years ago, there used to be a call list, literally before the internet.
There used to be a call tree, I remember when I first joined.
Yeah, I was on that too.
Rich, spaceweather.com.
Yeah.
Space Weather has a text you can get on your phone.
Yeah,
And
there's
you can
also,
configure it too.
there's also Aurora Forecast Pro or something like that that I have for $10 for lifetime that sends you a text message.
Most of the communication with the club members happens with email, which is really slow by today's standards. You know, there are far more efficient ways to communicate, but the best we've got right now is emails. I hope everybody was aware that this was going to happen. I watched spaceweather.com like all day long. When the KP index is up, the KP index was over 9 for a long, long time that night.
Well, I mean, they had predicted this stuff too.
Yeah.
So we were waiting for it.
Yeah. So it was a night to remember for sure.
Yeah.
Like, it's been about at least 25 years since I've seen an aurora like this,
Yeah.
probably longer.
So you mentioned the nightscape, so that photo, the first photo, that was one of about 1,300 shots that I did
Wow.
that night. And this, I have a video, I'm developing a video on it.
Oh, time lapse.
That was all nightscape.
Nice.
And that was with a Canon 6D modified. So the reds came out a little bit more, but the reason, I sent something out, the reason the reds came out was because the reds are the highest one, 200 to 300 kilometers high. And basically with the curvature of the Earth, a lot of us were just seeing just the red.
That's right.
Yeah.
As a matter of fact, the next two slides are from Southern folks. Wait 1 second, Doug.
I ran a time-lapse video all night, so I have it end to end. Corey has a few short segments which he may
And maybe we could see that at the July meeting, right? Because are we going to do a member night at July meeting?
There's one more speaker slot for that.
Okay.
I have the time lapses loaded on this computer. I might play them after the meeting.
Okay.
Because we're really tight on time.
Well, this one was from our friend in North Carolina, Roger Ivester. They're North Carolina. You could see the tops of those aurora displays. They're really far away from North Carolina, but you've got those reds high up. And I don't know if that's light pollution down below, or if that's a little bit of green from aurora light down here, but you know, it looks aurora like to me. And there's one more shot from pretty far south in Mexico.
Wow.
So this aurora was almost seen literally around the world.
That's where I was for the eclipse.
That's right, that's where you were for the eclipse. Perfect.
That's wild.
So, like I said, that was a night to remember. I don't know when we'll get another display like that, but you know, maybe we can figure out a way to make it more efficient to communicate this sort of stuff with our members in real time.
When I saw it, I started calling people.
Yeah, oh, I did too.
Oh, my
Yeah,
neighbors.
sure.
I said, get up, get your ass outside.
There was a steady stream of people just coming up the hill. So you probably had, Al, you were up there. I don't know if anyone else here tonight was up at the clubhouse, but there was what, 20, 30 people that came up. They were annoying as heck because they wouldn't turn their headlights on.
Yeah, Diana Hatton was here earlier, and she observed from an area in Sudbury. She said there were tons of people that had just gone to this field to watch the event. Pretty cool.
That was fantastic.
I love that we get to see this stuff. So anyway, now the boring stuff, the planet roundup for the month. Really, the only planet that's still easy to see is Saturn, but you still got to get up pretty early to see that. Transits at about 7 in the morning. That's after sunrise, of course. Ring tilt is less than 3 degrees now. So it's a unique opportunity to see Saturn. Ring plane passages are going to occur in 2025. But right now, the rings are these two little, almost like V-shaped structures that stick out. They're like little beams that stick out from the edge of the disk. Jay, you have a question?
Yeah, during July, there's going to be quite a few transits
of
Saturn.
Of Titan?
Titan and Tethys.
Okay, Titan's a big guy. Yeah. So I was going to mention that. So if you've got a software program like Sky Safari Pro, that's my favorite, Sky Safari. You can just put it on Saturn, center it, lock it in place, and run time forward. You can see every 16 days, Titan's going to start to do occultation, transits across the disk of Saturn. And it's a big move. So you'll be able to see those really well from the Earth. Next year, Saturn will, there'll be shadow transits of Titan, across the disk. And it drops a big black shadow on the disk of Saturn. And we get to only see these things once every 15 or so years. Right? That's how long it takes between ring plane passages. And since you're looking at the plane of the Saturnian system, now it's in line with the sun, so these moons are casting shadows onto the disk of Saturn. So it's really cool.
Those Titan transit starts tomorrow morning right after sunrise.
Tomorrow morning, the first Titan transit.
Approaches it.
You're going to grab that?
And it goes, going to be going, I'm going to take a look just to see how well it shows up after sunrise. We've got another one on June 30th that occurs, I think we get to see it cross the disk about a third of the way across the disk in a dark sky.
Yep.
You'd have to get up and look, probably around 3, or 2 or 3 o'clock.
If you're interested in seeing those, I mean, I can, just like I do with my Jupiter stuff in the fall, I can send out an email with the transits of Titan. They're kind of cool to watch. Anyway, for the rest of it, Mercury and Venus, unobservable right now. That's even a little too close for me with Venus. Jupiter and solar conjunction was only on May 18th, still only 16 degrees from the sun. Neptune and Uranus also in the morning skies. The only planet in the evening skies technically is Venus, just passed a solar conjunction just a few days ago, when it was actually eclipsed by the sun. Making it totally unobservable. Next slide. So again, the variable star of the month is T Corona Borealis. All this hype about T Corona Borealis. T Corona Borealis last erupted in 1946. And the time before that was 1866, giving it about an 80-year period between these eruptions. Now, as sort of a scientist, that doesn't seem like a big trend to me. I don't know that I'm going to, that's just basically, this could be just noise in the just grand scheme of things, right? But if it lives up to its expectations, they, whoever they are suggesting that this will go into eruption during the summer. And the reason, if you really want to see this, it's almost like a once-in-a-lifetime chance, right? 80 years, right? They're pretty few and far between. It's relatively easy to find. There's Corona Borealis, and the star is expected, T is right here. Right now it's running visually about 10th magnitude. It's expected to reach the brightness of Gamma here. This is about a 2nd magnitude star. And so if you want to see this thing, here's another reason to do your homework and pay attention. So the next slide is the light curve from 1946. And I put that red line on there at apparent magnitude of 5. Now most of us do not enjoy magnitude 5 skies, right? My sky in Framingham is getting worse and worse by the minute. It's about minus 4.3. The reason I put that line in is, look at how little time that star spends above 5th magnitude. It's not long. So when it goes into eruption, you probably have like a week or so to look for it before it drops right back down again to, beyond what we could see with our naked eye. And so that's a good reason to pay attention. I look at the AVSO website every morning to see what last night's magnitude estimates were. And the moment that thing starts to change, I'll light up the email with it's up, get out, and observe it. So I'll probably keep this as the variable star of the month until it does something. It could take decades for all we know, right? And I'll throw this out to you. 80 years before 1866 was 1786, right? Do you think William Herschel would have missed this? I don't think he would have missed it. So I don't know if this 80-year thing is real or if it's just me.
The pre-eruption dip is occurring right now.
It's happening right now, yes. Yeah. And that's the greatest observation to make us think it's about to do it again. So keep an eye on T Corona Borealis.
Yeah, but if you look at that, that means it'll be several months.
Right. And they're talking about between now and September. But we don't really know. Anyway, they're pretty fun. It'll be fun to watch when it happens. But again, you got to catch it because it's up and down quickly. All right, so
So
next.
why isn't it 2026 that it's gonna happen? 80 years.
You're right, what Mario just said, that
Now we're seeing it.
dip is suggesting that it's on the verge of going. But again, do you think William Herschel would have missed this 80 years before, 3 eruptions ago? I don't think he would have missed it. And I don't see anything in the literature that suggests that this has been an ongoing every 80 years sort of thing. We'll see.
I searched on the Sky National website. It seems that there's an article about it in 2016. So, like, since 2016, have they been looking for it?
Yeah, they've been watching this for a while. That's a Bob King article. I just read that the other day. It's pretty good. Again, as far as this eruption goes, I didn't think Shoemaker Levy was going to put a dent in Jupiter at all. What do I know? Anyway, the variable star of the month is Ras Al Jethii, is Alpha Hercules. It's a beautiful double. Struve 2140. 3.4 and 5.4, spectral class M5 and G5. It's a beautiful double star. Easy to find. Check it out. All right, that's the double star of the month. And the observer's challenge of the month is NGC 6118, alleged to be the most difficult object in the Herschel 400 category. Doug Paul, you had suggested after imaging it that this was going to be really challenging for visual observers. So try it, see what you can see.
Does that mean imaging it is off limits?
No, a lot of the imagers will submit reports to Roger and Sue French. Sue French also helps him with it. You see, there are a lot of people that do it visually. There are a lot of people that do images. So, but a lot of I mentioned it in the Observer's report for the year, a lot of our members are still actively involved in monthly reports to Roger and Sue. So kudos to us. Because we're by far, of all the clubs in the world, we're the ones that submit the most reports to Roger
Somebody
and
just
Sue.
commented on his mailing list, how come so many of the reports come from Massachusetts?
Right, because we're awesome, that's why. That's why. All right, next slide. So here's a picture from Doug Paul of that pesky little galaxy. Doug, any?
Yeah, that's 8 hours with a 6-inch aperture.
8 hours?
Yes, that's 2 to 4 times more time than I usually put in on these objects.
Yeah.
The center isn't that hard to see, but to get the spiral portion, that really took all the data.
Yeah, we'll see how it comes out. Mario, I think your picture's next, your image is next. Whoa, there's Mario's. With 30, is that with the 32?
Yeah, well, I have a little more aperture, so. I didn't have to do 8 hours. But May was an awful month, wasn't it?
Yes.
So I did, and it doesn't rise above the horizon for all that long, so I concentrated on getting red, green, blue. And by the time I was done with green, it was starting to get too low. So I combined that, there was no other clear nights, with an image I took of this, it was 4 or 5 years ago. And through the marvels of Fix Insight, I was able to combine them, and that's a combined probably about 3.5 hours of luminance from 2019 and red, green, blue from just a few weeks ago.
Okay.
What monochrome camera were you using?
I use a ZWO 6200.
Okay.
So get out there and try and see it. It's fun.
The colors are very subtle on that one.
Okay, yep. For the next month, July, so a little planetary in Hercules, 6058. It's relatively small and relatively faint, but it's actually pretty easy to find. You just have to go look for it.
It's very bright.
Is it a bright one?
Yeah.
I mean, I've looked at it once in my whole life. So, okay, but it's relatively easy. It may look off the beaten path, and it certainly is overshadowed by M13 and M92. If it weren't for M13, M92 would be like a showpiece glide. It's beautiful.
Yeah, it's very compact, so it's very bright.
Check that out.
I did narrow band, and I had to do short exposures.
Yep. But
Yeah, I had to do short exposures as well.
6058, so check it out. It's pretty cool. All right, what's next?
And that image on the right there is a wide field.
Oh, okay. Okay.
Close up.
Perfect, thank you for submitting that. So our observing schedule for into July is end of the month in June, June 28th and 29th, July 5th and 6th, and July 26th and 27th. And like Steve mentioned, if you want to learn how to use the 25-inch, it's just a Dobsonian. It's easy to learn how to use the 25-inch. And so if you're interested in playing with that telescope, come on up, we'll show you how to take the covers off, and kick it, and make it go.
So with that, we'll close the meeting. Our next meeting will be July 11th. It is voted on. We skip August for any new members. We do not hold an August meeting. And we'll return in September. And just a reminder on that board meeting next week, or next 2 weeks, June 27th. And that'll officially close the meeting, but I'm gonna play some Aurora videos here if you like, so you're free to stand. We'll bring the lights up a little bit. There's refreshments at the back. Thank you all.